Okay, let's unpack this. Imagine trying to copy a massive instruction manual, say, one with 3 billion pages perfectly, page for page, billion by billion, with almost zero errors. And you have to do it faster than you can blink every single time. That's essentially what your cells are doing every day with DNA replication, duplicating the human genome, with its over 3 billion base pairs. The sheer volume of information and the precision required, it's just mind-boggling.
It truly is. What's fascinating here is that without this extreme precision, genetic continuity would be impossible. Think about it. Even a seemingly small error rate, like 1 in a million base pairs, would still lead to thousands of mistakes during each replication cycle of your genome.
Wow, thousands.
Yeah, but here's the beautiful paradox. While the system is incredibly robust and accurate, it's also, well, imperfect enough to allow for the genetic variation that drives evolution. It's a fundamental balance that underpins all life.
And that balance, that incredible process, is exactly what we're diving into today. You, our listener, shared an in-depth chapter from Essentials of Genetics, and our mission is to distill how DNA copies itself, the intricate dance of molecules involved, and why this fundamental process is so critical to life, health, and disease. We'll try to make it engaging and easy to understand, tracing this incredible journey all the way back to Watson and Crick's initial groundbreaking insights. So our story really begins with that pivotal moment in 1953. James Watson and Francis Crick published their classic paper announcing the double helical model of DNA. It was an instant aha moment because they immediately recognized that the specific pairing of A with T and C with G nucleotides, it implicitly suggested a copying mechanism. It was almost staring them in the face.
Indeed. Their insights sparked a pretty fierce debate among scientists about how this copying actually occurred. They considered three main theoretical modes of replication, each with vastly different implications. The first, and the one Watson and Crick proposed, was semiconservative replication. Imagine a zipper. You pull it apart, separating the two original strands. Then, on each of those separated halves, you build a brand new complementary half. So each new DNA molecule is a hybrid, one original old strand and one newly synthesized new strand.
Okay, like one new in each daughter molecule. Makes sense.
Exactly. The second idea was conservative replication. Here, the original double helix would somehow remain entirely intact, acting purely as a template, and an entirely new, pristine double helix would be synthesized from scratch. It's like photocopying a book and getting two completely separate copies. The original, untouched, and a brand new one.
Right, the original just sits there.
And then there was dispersive replication. This was the most complex to imagine, really. The parental DNA strands would somehow be chopped up, and these pieces would intersperse with newly synthesized DNA in both new helices, resulting in a patchwork of old and new DNA throughout each strand.
That sounds messy.
Very. Thankfully, that isn't what happens.
Okay, here's where it gets really interesting. How do you prove which one of these, kind of wild ideas is actually happening inside a cell? Enter Matthew Meselson and Franklin-Stahl in 1958 with their absolutely elegant experiment using bacteria.
Oh, it's a classic. Beautiful work.
They had this brilliant idea: what if they could label the old DNA and track it? They grew E. coli cells for many generations in a medium containing a heavy isotope of nitrogen, N15, which incorporates directly into the DNA, makes it denser. Then they transferred these N15 labeled cells to a light N14 medium and allowed them to replicate just once.
Right, so you start with heavy DNA, then let it copy itself using light building blocks.
And to distinguish the DNA, they used an ingenious technique called sedimentation equilibrium centrifugation. Imagine a test tube filled with a special liquid that gets denser as you go from top to bottom. When you spin the DNA samples in this liquid at high speed, they settle precisely at the level where their density matches the liquid, like finding its own buoyancy level.
Exactly. Heavy sinks lower, light stays higher.
So after one generation in the N14 medium, they observed something remarkable. The isolated DNA was present in only a single band of intermediate density. Not heavy, not light, but right in the middle.
That single intermediate band was incredibly powerful evidence. It immediately ruled out conservative replication, because if that were the case, you'd see two distinct bands, right? One heavy band, the original DNA, and one light band, the completely new DNA.
Got it. So the intermediate band means the new DNA molecules must be hybrids.
Precisely. It strongly suggested semi-conservative replication, where each new molecule was a hybrid of one old N15 strand and one new N14 strand.
But they weren't done. What happened in the next generation if they let them divide again?
Good question. After two cell divisions in the N14 medium, the DNA samples showed two distinct bands. one intermediate band like before, and a brand new lighter band corresponding to purely N14 DNA.
Ah, okay. So the hybrids replicated again, making more hybrids and some completely light molecules.
Exactly. This result perfectly aligned with semiconservative replication. The hybrid molecules from the first generation would again replicate, producing half hybrid and half light molecules.
So that confirmed semi-conservative replication for bacteria, right? But how did they completely rule out that patchwork idea, dispersive replication? Couldn't that also maybe give an intermediate band?
That's a crucial point. They took an extra step to address that. They isolated those hybrid molecules from the first generation and gently heated them. Heating separates the DNA double helix into single strands. Now, if replication were dispersive, those single strands, being a patchwork of old and new pieces, should still be of intermediate density, mixed heavy and light along the strand. But what they observed were distinct heavy N15 single strands and light N14 single strands, not an intermediate mix.
Ah, so the strands themselves were either all old or all new.
Correct. This, combined with the two-band result in the second generation, conclusively proves semi-conservative replication. The broader significance, it was just a landmark experiment in molecular biology, directly confirming Watson and Crick's initial hypothesis with such elegant precision.
And it wasn't just bacteria. Just before Meselson and Stahl's work, in 1957, J. Herbert Taylor, Philip Woods, and Walter Hughes had already provided compelling evidence for semi-conservative replication in eukaryotes, too. They used root tips of the broad-bean Vicia faba good dividing cells and labeled the DNA with radioactive thymidine. Then they used autoradiography to see where the radioactivity went.
Right, using radioactivity as a label instead of isotopes.
Their results showed that after one replication cycle with the label, both sister chromatids of the chromosome were radioactive. Then, after a second replication cycle in unlabeled medium, only one of the two sister chromatids of each chromosome was radioactive.
Which is exactly what you'd expect with semi-conservative replication. Each chromatid after round 1 is a hybrid. After round 2, you get one hybrid and one unlabeled chromatid pair. So this experiment really cemented the universality of semi-conservative replication across all life, from simple bacteria to complex eukaryotes. It was a critical validation of the Watson and Crick model.
Okay, so now that we know how DNA is copied semi-conservatively, let's dive into the who and what. The amazing enzymes and proteins that make it all happen. We'll start with bacteria, as they're often our best studied models for these kinds of intricate processes.
Good starting point. Simpler system, relatively speaking.
Right. Every replication journey begins at a specific spot, where DNA is actively replicating, the helix unwinds, forming that Y-shaped structure called a replication fork. And a replicon is simply the unit of DNA replicated from a single origin.
And John Cairns' work in E. coli was foundational here. He showed that bacteria typically have a single origin of replication called oriC in E. coli. And their entire circular chromosome is essentially one replicon.
Okay, one starting point for the whole circle.
From oriC, replication proceeds bidirectionally. That means two replication forks move out in opposite directions. They zip around the circle and eventually merge on the other side, completing the duplication.
Makes sense for a circle, but who does the actual building?
The initial insights into the actual machinery came from Arthur Kornberg in 1957, a huge achievement. He isolated the first enzyme capable of directing DNA synthesis in vitro from E. coli. He named it DNA polymerase A-IR. He showed it needed all 4 DNTPs, the building blocks, and a DNA template to copy. And critically, he discovered that polymerase nucleotides only in a 5 to 3 foot direction, building onto an existing 3-OH group.
So when Kornberg found DNA polymerase I, that must have seemed like the answer, right? The enzyme that replicates DNA.
That's a fantastic point, because for a while it was considered the main player. It made sense, it did the job in the test tube. But then a really clever experiment in 1969 with a mutant bacterium called pol-A1 threw a huge curveball.
Oh, what happened?
This mutant lacked functional pol-1. It couldn't make the enzyme properly. But it still replicated its DNA and reproduced successfully.
What? How?
Exactly. However, these cells were severely deficient in their ability to repair damaged DNA. This crucial observation indicated that while pol-vientis is involved in DNA synthesis, its primary role in vivo in the living cell is actually in DNA repair.
Ah, okay. So it's more like a handyman than the main construction crew.
That's a good analogy.
Yeah.
The real workhorse for the bulk of DNA replication turned out to be DNA polymerase III, which was discovered later. Other polymerases like Pol-Theta-4 and V were also found, and they're primarily involved in various aspects of DNA repair, especially under stress conditions.
So DNA pull-through is the star of the show for actual replication, and you said it's not just one simple enzyme, right? It's a holoenzyme.
That's right. It's a massive molecular factory, really complex, made up of many different polypeptide subunits. You can think of it as having several crucial parts. There's the core enzyme, which has subunits that one rapidly builds the new DNA, that's the 5 to 3 foot polymerization, and another acts as an immediate vital editor. It proofreads every single nucleotide added to catch errors on the fly.
The proofreading part, super important for accuracy.
Absolutely essential. This build and edit team is what makes replication so incredibly precise.
But there's more to it than just the core, you said.
Yes. To really get its work done efficiently, Pol III needs help. There's a group of 5 other subunits that form what's called a sliding clamp loader. This loader complex uses ATP energy to load another crucial component onto the DNA.
Which is?
The sliding DNA clamp. It's this donut-shaped protein, the beta subunit in E. coli. It literally encircles the DNA strand like a ring in a finger. And by physically linking to the core enzyme, it tethers Pol III to the DNA.
Okay, so it keeps it from falling off.
Exactly. It vastly increases the processivity of the polymerase. Processivity just means how long the enzyme stays attached to the template and synthesizes DNA before detaching. Without the clamp, Pol III would only add a few nucleotides and fall off. With it, it can add thousands, even millions. This is absolutely key to its speed and efficiency.
Wow. Okay, so it's a complex machine, but replication isn't just building, right? You mentioned unwinding, tension.
Precisely. DNA replication is far more than just adding nucleotides. It's a symphony of coordinated events. At the replication fork, there are about 7 complex issues that must be resolved simultaneously.
7. Okay, let's break them down.
First, the DNA helix must be unwound. You can't copy it if it's zipped up. This starts at specific sequences in E. coli's oriC. These are often rich in AT base pairs, which only have 2 hydrogen bonds, making them easier to pull apart than GC pairs, which have 3.
Easier to melt. Okay.
The DNA initiator protein binds to specific sequences within oriC and uses energy to begin destabilizing the helix, creating a small bubble. Then DNA helicase, often called the unzipper enzyme like DNB in E. coli, comes in. It uses ATP energy to power its way along the DNA, continuously unwinding the double helix and separating the 2 strands, expanding that replication bubble.
But wouldn't those single strands just want to snap back together immediately?
Good point. They absolutely would. To prevent this, reannealing single-stranded binding proteins, or SSBs, quickly coat to separated strands. They keep the strands apart and protected until they can be used as templates.
Okay, so unwind with helicase, keep apart with SSBs. What's issue number 2?
Number 2 is reducing the coiling tension, or supercoiling. As that helicase unwinds the DNA locally at the fork, it creates immense torsional stress further down the helix. Imagine trying to quickly unwind a tightly coiled rope. The part ahead of where you're unwinding gets even more twisted.
Right, it would get knotted up.
Exactly. This supercoiling could literally stop replication cold. To combat this, an enzyme called DNA gyrase comes into play. It's a type of DNA topoisomerase. Gyrase makes temporary cuts, either single or double-stranded breaks in the DNA backbone ahead of the fork. This allows the coiled strands to swivel around each other and relax, relieving that tension. Then, crucially, gyrase reseals the strands. It's all powered by ATP.
Wow. Cutting and resealing DNA to relieve tension. Incredible. Okay, issue 3.
Issue 3, and perhaps the big paradox we hinted at earlier, DNA polymerases can only add nucleotides to an existing strand. They need a free 3' group to attach the next nucleotide to. They can't start a new chain from scratch on a bare template.
Right. So how does the very first nucleotide get added, or how do they start those Okazaki fragments we'll get to?
That's where an ingenious, slightly counterintuitive solution comes in. RNA serves as the primer.
RNA, not DNA.
Yep. An enzyme called primus, which is essentially a specialized RNA polymerase, synthesizes short segments of RNA, typically 10 to 12 ribonucleotides long. These RNA primers are complementary to the DNA template strand. This provides that crucial free 3' group that DNA polymerase III needs to start adding DNA nucleotides.
So it lays down a little RNA runway first.
Exactly. And this is a universal phenomenon across pretty much all organisms. RNA priming is absolutely essential for initiating DNA synthesis. Once that RNA primer is in place, DNA polymerase III can latch on and begin synthesizing the actual DNA strand. That RNA primer will then have to be removed later, of course.
Okay, RNA primers. Got it. What's number 4?
Number 4 arises directly from the structure of DNA and the limitation of DNA polymerase. The two DNA strands of the helix are anti-parallel. One runs 5'3" model, and the complementary strand runs 3'5" Panama. But as we said, DNA pull-through only synthesizes in the 5'3" put direction.
Right, only one way.
So as the replication fork moves forward, let's say left to right, synthesis along one template strand can proceed continuously towards the fork. This is the leading strand. It just needs one initial primer, and then pull the third can go.
Smooth sailing on that one.
Pretty much. But on the other template strand, the one running 5'3" minutes towards the fork, synthesis must proceed in the opposite direction, away from the fork, to maintain that 5'3" minute synthesis rule. This means synthesis on this strand, the lagging strand, is discontinuous. It has to keep starting over. It occurs in short segments. Primus makes an RNA primer. Pull the third synthesizes a short stretch of DNA moving away from the fork. Then it detaches, moves back closer to the fork where new template has been unwound, waits for another primer, and synthesizes another segment.
And these short segments are the famous Okazaki fragments.
Exactly. Named after Tsuneko and Reiji Okazaki, who discovered them. These are short DNA fragments, about 1,000, 2,000 nucleotides long in bacteria, each beginning with its own RNA primer. Because of this continuous synthesis on one strand and discontinuous on the other, the overall process is often called semi-discontinuous synthesis.
Leading and lagging strands, Okazaki fragments. Makes sense. So issue 5 must be dealing with those RNA primers, right?
You got it. Issue 5 is removing the RNA primers, and issue 6 is filling the gaps left behind and joining the fragments. After the Okazaki fragments are synthesized on the lagging strand, and after the initial primer on the leading strand is synthesized past, DNA polymerase I steps back in. Remember its repair function?
Yeah, the handyman enzyme.
It uses a special activity. It has a 5' to 3' exonuclease activity to basically chew away the RNA primers from the 5' end. As it removes the RNA nucleotides one by one, it simultaneously uses this polymerase activity to fill the resulting gap with DNA nucleotides, using the adjacent Okazaki fragment's 3' end as a starting point.
Okay, so pol I cleans up the RNA and patches with DNA, but there's still a little break, isn't there, between the DNA patch and the next fragment?
Exactly. Pol I can't make that final connection. It leaves a tiny nick in the sugar phosphate backbone, a missing phosphodiester bond. That's where issue 6 is resolved by the molecular glue, an enzyme called DNA ligase. DNA ligase specifically catalyzes the formation of that phosphodiester bond, sealing the nicks and joining the Okazaki fragments into a continuous strand. We know how critical ligase is because in mutants that lack functional ligase, you see an accumulation of these unjoined Okazaki fragments. It's essential for completing the lagging strand.
Okay, unwind, stabilize, relieve tension, prime, synthesize continuously and discontinuously, remove primers, fill gaps, ligate. That's 6. What's the 7th crucial issue?
The 7th issue brings us back to accuracy, proofreading, and error correction. While DNA polymerases, especially pol third, are incredibly accurate, they're not perfect. They do occasionally insert the wrong nucleotide, maybe one in every 100,000 bases.
That sounds pretty good, but over billions of bases.
It adds up to a lot of potential mutations. So what protects us? DNA polymerase III, Ni, possess another vital enzymatic activity, a 3' for 5' exonuclease activity. This works in the reverse direction to synthesis.
Okay, how does that help?
This allows them to perform proofreading on the fly. If the polymerase adds an incorrect nucleotide, the enzyme can sense the mismatch. It often causes a slight distortion in the helix. When it detects this, the polymerase pauses, reverses course, just one nucleotide, using its 3' 5 exonuclease activity to snip out the wrongly incorporated base. Then it resumes synthesis in the forward 5' to 3' direction, inserting the correct nucleotide this time.
Wow, it checks its own work immediately.
It does. This vital proofreading mechanism increases the overall fidelity of DNA synthesis by about 100-fold, catching most errors as they happen. It reduces the error rate down to something like 1 in 10 million bases.
Incredible self-correction.
It's a key reason why replication is so accurate.
Okay, putting it all together then. The bacterial replication fork is this amazing coordinated machine, sometimes called a replicon, right? You've got helicases unwinding, SSBs stabilizing, gyrase relieving tension, primus laying down RNA primers, pol third extending DNA on both leading and lagging strands with its clamp, pol cleaning up primers and filling gaps, and ligase sealing the deal, all while pol third is proofreading.
It's an incredibly synchronized and efficient process, a true molecular machine.
So if bacterial replication is this complex with all these moving parts, What happens when you scale up to eukaryotes? We have vastly more DNA. It's packed into linear chromosomes, not circles, and it's wrapped up in those complex structures like nucleosomes. It sounds like a whole new level of challenge.
It definitely is. But let's establish the fundamental similarities first, because many core principles hold true. Eukaryotic DNA replication is also semiconservative. It involves origins and replication forks. It proceeds bidirectionally. It utilizes leading and lagging strands with synthesis always in the 5 to 3' direction. And the fundamental requirements for DNA polymerases, a template, dNTPs, and a primer remain the same.
Okay, the basics are conserved, but the differences must be significant.
They are. First, the sheer scale. Eukaryotic genomes are vastly larger than bacterial ones.
Yeah.
And surprisingly, their core DNA polymerases work much slower than bacterial pull thought, maybe 25 times slower.
Slower, with more DNA to copy. How does that work?
To compensate for this slower speed and the immense amount of DNA, eukaryotic chromosomes don't have just one origin of replication. They have multiple replication origins, thousands of them. For instance, the human genome might have 25,000 or more origins. Replication initiates at many points along each chromosome simultaneously. These origins fire throughout the S phase of the cell cycle, creating numerous replication bubbles that eventually merge. This parallel processing allows the entire genome to be copied within a reasonable time frame, despite the slower polymerase speed. Okay. Many starting points make up for the slower pace. Makes sense. What else is different? Eukaryotes also utilize a larger and more diverse cast of DNA polymerases. Humans, for example, have at least 14 different types identified, though many are involved in repair. For the main job of replicating nuclear DNA during S phase, 3 are primary players: pol alpha, pol delta, and pol epsilon. Okay, alpha, delta, epsilon, what do they each do? Pol alpha is unique. It actually has primase activity built into one of its subunits, so it synthesizes the RNA primer. Then, its polymerase subunit adds a short stretch of DNA nucleotides, maybe 10 to 20, right after the primer. But pol alpha has relatively low processivity. It falls off easily.
So it starts things off but doesn't do the bulk synthesis.
Exactly. After pol alpha lays down that initial RNA-DNA hybrid primer, an event called polymerase switching occurs. Pol alpha dissociates, and the other, more highly processive polymerases take over. Pol delta, poly, is generally thought to be the main polymerase synthesizing the lagging strand, and pol epsilon, pol A, is thought to primarily synthesize the leading strand. Both pol delta and epsilon are highly processive. They have their own sliding clamps, analogous to the bacterial one, and crucially, they both have that vital 3 to 5 bit proofreading exonuclease activity for accuracy.
So a switch from the starter polymerase alpha to the main workhorses, delta and epsilon. And are the Okazaki fragments different?
Yes. Another difference is that eukaryotic Okazaki fragments are much smaller than in bacteria, typically only around 100 to 150 nucleotides long.
Interesting. And what about the fact that eukaryotic DNA isn't just a naked strand in the nucleus, it's packaged up?
That's perhaps the most profound difference. Replication through chromatin. Eukaryotic DNA is tightly wound around histone proteins to form bead-like structures called nucleosomes. These are then further coiled and folded into chromatin, a highly organized packaging system. This means the replication machinery can't just access bare DNA. It has to contend with these histone roadblocks.
So how does it get through? Does it have to dismantle the chromatin?
Essentially, yes, but temporarily. Nucleosomes must be disrupted or slid aside just ahead of the replication fork to allow the polymerases and other machinery to pass through. And then, almost immediately behind the fork, the nucleosomes must be rapidly reassembled onto both of the newly synthesized daughter DNA molecules. This ensures that the chromatin structure, which is important for gene regulation, is faithfully inherited.
Wow, that sounds complicated. Are there special proteins for that?
There are specialized proteins called chromatin assembly factors, CAFs, escort histones and help deposit them onto the newly replicated DNA. And importantly, the synthesis of new histone proteins is tightly coupled to DNA synthesis during the S phase of the cell cycle, ensuring there are enough new histones to package the duplicated genome.
Okay, multiple origins, different polymerases, smaller Okazaki fragments, and replicating through chromatin. That's a lot of extra complexity.
It is. But there's one more major challenge unique to eukaryotes.
Which comes from the fact that their chromosomes are linear, right? Not circles like bacteria. They have ends.
Precisely. The final layer of complexity in eukaryotes comes from their linear chromosomes. Those ends, the telomeres, present unique challenges for the standard replication machinery.
I always found the end replication problem to be one of those elegantly simple challenges with a surprisingly complex solution. How exactly did scientists first even realize the ends of chromosomes were a problem?
It stems directly from the mechanism of lagging strand synthesis and the need for RNA primers. Think about the very end of the lagging strand template. When the final RNA primer is removed from the 5-foot end of the newly synthesized strand, there's no upstream DNA strand with a 3OH group for DNA polymerase to extend from to fill that gap.
Ah, right. The polymerase needs something before the gap to build onto. At the very end, there's nothing before it.
Exactly. This means, with each round of conventional DNA replication, the 5-foot end of the newly synthesized lagging strand would be shorter than its template. If this happened repeatedly over many cell divisions, the chromosomes would progressively shorten from both ends, eventually leading to the loss of vital genetic information. It's like a ticking clock, if you will.
A built-in shortening mechanism. Not good.
Not good at all. The other issue is that these bare, linear DNA ends could potentially be recognized by the cell's machinery as double-stranded breaks dangerous DNA damage. This might trigger DNA repair mechanisms that could mistakenly fuse chromosome ends together, leading to genomic instability, or trigger pathways that lead to cell death or senescence.
Okay, so chromosomes would shorten and potentially look like damage. How do cells solve this?
The solution involves specialized structures at the chromosome ends called telomeres. Telomeres are essentially protective caps made of repetitive DNA sequences. In humans, for example, the sequence is 5-foot T tag G3 bucks, repeated hundreds or thousands of times. These repetitive sequences don't typically encode proteins. Their job is structural.
Just buffers at the end.
Buffers, yes, but they also form unique structures. One strand, the G-rich strand, extends beyond the C-rich strand, creating a 3-foot G-rich overhang. This overhang can actually loop back and tuck into the double-stranded region of the telomere, forming a protective structure called a P-loop. These T-loops, along with a dedicated complex of proteins called the shelterin complex that binds specifically to telomeric DNA, help shield the chromosome ends. They protect them from degradation and prevent the cell from mistakenly identifying them as broken DNA.
Okay, so telomeres act as caps and hide the very ends. But that doesn't solve the shortening problem itself, does it? The buffer would still get worn down eventually?
Correct. The structural protection is one part, but the ultimate solution to the shortening problem is an incredible enzyme called telomerase.
Ah, yes. This was famously discovered by Elizabeth Blackburn and her graduate student, Carol Greider, in studies of the ciliated protozoan tetramina, a Nobel Prize-winning discovery. They found this enzyme that could actually add those repetitive sequences back onto the ends.
It's a truly unique enzyme. Telomerase is a ribonucleoprotein, meaning it contains both protein and RNA. The protein component, called TERT, is a reverse transcriptase, an enzyme that can synthesize DNA using an RNA template. And the crucial RNA component, called TERK, is embedded within the enzyme itself.
So it carries its own template.
Exactly. The TERK RNA serves a brilliant dual role. Part of it acts as a guide to recognize and bind to the G-rich overhang of the telomere. And another part of the RNA serves as the actual template for the TERT protein to synthesize new telomeric DNA repeats, extending that 3-foot G-rich overhang.
Wow. So it binds the end, uses its internal RNA to make more DNA repeat, then scoots over and does it again.
That's the essence of it. Telomerase binds, extends the 3-minute overhang using its RNA template through reverse transcription, then it translocates or repositions itself and repeats the process multiple times, adding many copies of the TTG sequence. Once this 3-minute overhang is sufficiently elongated, the cell's conventional replication machinery primus, DNA polymerase, probably Pol alpha and delta, and ligase can then synthesize the complementary C-rich strand, filling in most of the gap. The end result is that telomere length is maintained, counteracting the shortening that would otherwise occur with each replication cycle.
An ingenious solution to the end replication problem.
Absolutely. Now, zooming out, the implications of telomeres and telomerase for human health and disease are really profound.
That's so...
Well, in most of our normal somatic cells, the regular cells making up our tissues, the gene encoding telomerase, is turned off or expressed at very low levels. As a result, their telomeres do shorten with each cell division. This progressive shortening acts like a cellular clock. Eventually, the telomeres become critically short, triggering a state called senescence, where the cells permanently stop dividing, or sometimes triggering apoptosis, programmed cell death. This is thought to be a protective mechanism against uncontrolled proliferation like cancer, and might contribute to the aging process.
So most of our cells have a limited number of divisions because their telomeres run down.
That's the general idea for many cell types. However, some cells need to divide indefinitely, or for very long periods. Think about embryonic stem cells, which have to generate an entire organism, or adult stem cells that replenish tissues throughout life. or highly proliferative cells like activated immune cells. These cell types do express telomerase, allowing them to maintain their telomere length and retain their capacity for long-term division.
Okay, so telomerase activity is crucial for stem cells and highly dividing cells. What happens when it goes wrong?
We see the impact clearly in certain diseases. For example, dyskeratosis congenita, DKC, is a rare genetic disorder caused by mutations in the genes for telomerase components, like TERT or TERK, or in shelterin proteins. Patients with DKC have abnormally short telomeres, leading to premature failure of tissues that rely on cell renewal, like bone marrow failure, skin abnormalities, and an increased risk of cancer. It essentially looks like premature aging in certain systems.
A direct link between telomere maintenance failure and disease.
Yes, and the connection to aging itself is an active area of research. While it's complex and debated, there's a general correlation observed between shorter telomere length and increased age or age-related diseases. Factors like chronic stress, inflammation, and poor diet have also been linked to accelerated telomere shortening. And quite fascinatingly, some studies in mice engineered to age prematurely due to telomere dysfunction showed that reactivating telomerase could actually reverse some aging symptoms, like tissue atrophy and even neurodegeneration.
Wow, reversing aging symptoms in mice by fixing telomeres. That's provocative. But perhaps the most intriguing connection, and the one with maybe the biggest clinical implications, is with cancer, right?
Absolutely. Here's the critical link. While most normal somatic cells lack telomerase activity and eventually senesce, the vast majority of human cancer cells, maybe 85–90%, have found ways to reactivate or maintain telomerase expression.
So they overcome that limit on cell divisions.
Exactly. This allows them to maintain their telomere length despite undergoing countless rounds of cell division. This effectively grants them replicative immortality, which is one of the key hallmarks of cancer, enabling the uncontrolled proliferation that forms tumors.
So cancer cells turn telomerase back on to keep dividing forever.
That's the prevailing model. This critical difference between most normal cells, telomerase off, and most cancer cells, telomerase on, presents a very attractive therapeutic target.
Because a drug that inhibits telomerase might specifically harm cancer cells.
Precisely. The idea is to develop anti-telomerase drugs that could block the enzyme's activity in cancer cells. This should, theoretically, cause their telomeres to shorten with each division, eventually leading to senescence or cell death, selectively killing the cancer cells or halting tumor growth, while having minimal effects on most normal tissues that don't rely on telomerase. Several such drugs targeting telomerase are indeed in various stages of preclinical and clinical development as potential anti-cancer agents. It's a very promising avenue of research.
A really clear example of how understanding a fundamental process like DNA replication can lead directly to potential new therapies.
Without a doubt.
So what does this all mean then? We've gone from Watson and Crick's initial flash of insight about the double helix suggesting a copying mechanism, through the elegant experiments proving semiconservative replication, to the incredibly intricate molecular machinery, the polymerases, helicases, ligases, the whole replicome working with astonishing speed and accuracy, dealing with challenges like chromatin and chromosome ends, and finally seeing the profound implications this has for aging, disease, and cancer through telomeres and telomerase. Our deep dive into DNA replication really reveals just how exquisitely designed yet adaptable life's most fundamental copying mechanism truly is.
It really is remarkable, and it raises, I think, an important question to ponder. We see this incredible precision in DNA replication and the multiple layers of repair ensuring genetic stability. Yet the system isn't perfectly perfect. It allows for some errors, some mutations to slip through, providing the raw material for evolution. So how much of our genetic destiny is truly fixed by the code we inherit, and how much remains a dynamic interplay, an interplay between that inherited code and the continuous, faithful, yet subtly adaptable process of its replication and maintenance throughout our lives?
A dynamic balance between stability and change encoded right into the copying process itself. That's definitely something to think about.
It's a question that continues to drive scientific exploration in genetics, aging, and disease research.
Keep exploring, keep questioning, and join us next time for another deep dive.